Good afternoon. I'm Nick Rishwain. I'm the Vice President of Experts.com, and we are here today with Suzanna Ryan, and she is a forensic DNA expert with over 15 years. It may be more than that now, but over 15 years of experience in forensic serology and DNA analysis. This blab is brought to you by Experts.com, and I'm going to ask Suzanna to give us a few bits and pieces about her experience, maybe elaborate a little bit on what forensic serology and forensic DNA analysis is. And after about 15 or 20 minutes, we'll see if some people want to come in and ask some questions after we have a chance to talk a little bit. Does that work for you?
Sounds good to me, yep.
All right, so tell us a little bit about forensic DNA analysis and forensic serology and what those two things mean.
Sure, okay, well, you can kind of think of it as two parts. The science is forensic serology to identify body fluids, and then DNA analysis to figure out, once you've found a body fluid, who does it belong to? So, when we talk about forensic DNA, that's the application of science to law, and most of the work that I do is criminal work. I do a little bit of civil work, but mostly criminal work. And so it is trying to find a body fluid or even skin cells. Now, the sensitivity has gotten so great with the testing systems that we can get DNA from skin cells. So once you find that body fluid, then trying to figure out if you can get a DNA profile, and then you compare that to a known reference sample, and that will help you try to figure out who it is.
Important to know that you have to have a reference sample in order to compare it to something. I'm assuming that would be Go ahead.
Yes and no. Yeah, yes and no.
Okay.
Typically, yes, you're going to have a reference sample, but then there is the CODIS system. That's the combined DNA index system. So let's say you have you find a DNA profile at a crime scene, and maybe you do have some suspects. You compare it to those suspects, but there's no match. Then the next thing that you can do is enter it into the database. So then you figure out if there's any convicted offenders, any individuals who have been arrested for a particular crime, see if there's a match in the database. So that would be the next step.
Okay. And it sounded like you were alluding to, I did a little bit of reading on your site and things like that before we got started, and I was interested. We're mostly people like myself who are essentially lay people when it comes to this type of thing. We understand a lot of the CSI and DNA stuff from TV shows. And you were, when we talked earlier, you had actually talked about the fact that I'm used to seeing that they use DNA sample in a homicide case or in a capital case, a major crime. And you mentioned something to the effect that this is also being used in lesser offenses more and more.
Absolutely, yes. So I would say probably within the past 5 to maybe 7 or 8 years, it has been much more common for DNA to be used in not just the serious cases like rapes and murders, things like that, but now we're seeing DNA evidence used in everything from burglaries, robberies, basically any sort of crime that you can think of. You can potentially get a DNA profile. And the reason for that is the type of testing that's used today is so much more sensitive. So, every year it seems like there's more increases in the sensitivity of the testing, whether it's the kit that is used to amplify or copy the DNA, or the instrument that's used to analyze the DNA. We're getting to the point now where we can get DNA profiles from just a few cells worth of DNA. So I see that as a good thing, but can potentially be, I don't want to say a bad thing, but you need to be careful and make sure that the DNA profile that you're finding at the crime scene is truly relevant to the crime and didn't just transfer there in some some other manner, because now we're becoming more and more aware that there's things like secondary transfer.
Yeah, you're bringing me, actually, you're transitioning into something I wanted to get into
Gotcha.
because in my reading, and in fact, you have an article on our site about transfer DNA.
Mm-hmm.
Can you explain that to us? What is transfer
Sure.
DNA, kind of for the layperson, or for how you would explain it to an attorney who is looking to hire your services?
Yeah, sure. Well, I mean, when we talk about transfer DNA, I mean, all DNA is transfer, right? Whether it's primary transfer, where even if it's a body fluid, if you're bleeding, then you leave DNA behind because your blood transfers to the knife or whatever. But we also have DNA in our skin cells. So a lot of times when we talk about transfer DNA, we're really referring to skin cell transfer, or DNA that's on the surface of our skin. So it'd be almost basically invisible DNA, right? You can't really see that. So primary transfer would be a direct transfer, whether it's a person to a person. If I shake your hand, there's a possibility that I could transfer some of my DNA to your hand, and just like you could transfer some of your DNA to my hand. Now, there's a lot of variables involved, whether you'll actually leave enough DNA to be detectable or not.
Okay.
But that possibility exists. So that would be a primary transfer.
All right. Okay.
Now, if you touched an object, you touch a coffee cup or something like that, same thing. You could transfer. There you go.
I'm wondering what I'm transferring here.
Yeah, all right. Yeah. Yeah. So you could transfer some of your DNA in that manner as well to that object, right? So that, again, that's primary or direct transfer.
Okay.
Now, what we have to be concerned about is secondary transfer or indirect transfer.
Right.
So in that first example, you and I shake hands, and now I've got your DNA on my hands, and then I pick up a gun or a knife or something used in a crime. It's the possibility, it doesn't always happen, but it's possible for your DNA to be transferred to that gun or knife, and you never touched it. So that's what I talk about when we have to be careful, where we're seeing the sensitivity of the testing increase, but you have to make sure that that is truly relevant to the crime and not just an innocent secondary transfer.
This may be too detailed for what we're doing here today. It may be for another blab in the future. But how can you distinguish between? So I shake your hand, and then I go out to the shooting range or something, and now there's some cells on my gun that belong to you. How do you distinguish in that situation? Is there? I imagine a lot of testing. Okay.
That's the problem. That's the issue. You can't really. So there's a lot of studies going on right now about transfer of DNA, because probably about 5 years ago, people would say, other experts even would say, well, it's possible, it might happen, but it's really not very likely. And now, again, with the increases in sensitivity, there's been additional studies done with the newer technology, and now we're seeing that it does happen. There was just a study out very recently, some individuals that, they did some testing on. Now, in this instance, they had people shake hands for 2 minutes. Normally, you wouldn't shake somebody's hand for 2 minutes, but they wanted to see some level of contact, how, that was what their cutoff was. And then they had the other person touch a knife, and they were seeing secondary transfer all the time. Oh, I can't say all, about 80% of the time. And the problem was, you can't always tell. I mean, there is no way through DNA analysis to tell whether it's primary or secondary. So in that instance, they saw all levels of transfer. They saw no transfer occurring. They saw mixtures where now, it'd be my DNA and your DNA on the knife if we shook hands and then I touched the knife. And they also saw instances where just your DNA transferred to the knife and none of mine. Yeah. So that was pretty surprising.
That's wild.
Yeah.
So I imagine then if we can't detect, why your DNA was on my knife or my gun, then would I be right in the assumption that, however, this has to do with establishing a reasonable doubt as to whether or not you were involved in a crime, or is that where that type of thing would be used? I was reading one of your very glowing testimonials on your page from a public defender who said that the transfer DNA was a big part of a case in Kern County.
Sure.
Is that what we're getting at? If we have multiple the DNA from multiple people on this weapon or something like that, then how can we be certain who was in possession of it? Is that
right? It seems like a lot of the times when I do testify, because I review a lot of cases and I don't always go and testify. A lot of times I say, yeah, it looks all right, or maybe there's a little minor error here and there, but overall looks fine.
Okay.
Sometimes when I testify, it is because there are possibilities of DNA transfer that have not been addressed. So, I think that's where investigations come into play, right? I think a lot of times people hear DNA and they think, oh, that's the silver bullet. His DNA is present. And then they don't want to look at any other like corroborating information or circumstantial evidence. And you have to look at that. You have to have the investigation aspect as well, to find out, was this person even in the same area at the same time? Is there a plausible explanation, or isn't there? Maybe there is no other explanation for their DNA.
Yeah. And in law school, we were taught that you build a case brick by brick, in evidence class, brick by brick, you sure. Pile up a little bit at a time. And I know that we have one attorney, Mitch Jackson, in the room with us right now. And we also actually have an investigator, Tamara Shelley. So when we get close, they probably would have some questions that they may want to ask and have some good content to provide.
Sure.
In your practice today, we were talking earlier, you spoke about a couple of things, and then I saw a couple of things on your website that I wanted to make sure and cover, because this really is about you and your services. I noticed that you do some educational seminars. The training is one of the things that you offer. Tell us about the training that you do, and who do you train?
Well, I've done trainings for attorneys, for both prosecution and defense. I've done trainings for defense investigators, for cold case investigators. I'm actually a member of the American Investigative Society of Cold Cases, which is a cold case group that reviews cases pro bono. So I'll just get a little information in for them, because it's a fantastic group. They've got amazing people involved in
and I'm going to post a link to, I think it's your presentation, the July 2015 presentation to the AISOCC. Is that them?
Yes, ASOC, yes.
Do you have a problem with me posting that down here so people can take a look at that?
No, that'd be
it's
fine.
On your website.
Yeah. That's fine. So that's recently I did give a presentation for them. Couple of presentations, and talking about DNA in cold cases, because there are some differences. When you have cases that are 10, 15, 20 or more years old, then you have to be aware that the way things were handled in the past, people didn't think about the fact that, well, if I put all of these items together, it's all the victim's clothing, so I can just put it all in one packaging. But now we know that there can be transfer of DNA from one item to another, even while it's packaged. And again, that goes back to the sensitivity of the testing today.
Yeah. Interesting that. Well, for many of us, our first real introduction to DNA was the O.J. Simpson case. And I was very young when that happened. As a teenager, but that was where we really got introduced to it, and they were able to poke holes in DNA at that time because it was such a new. I guess the technology was supposedly so new, the technology that they were using, the science that they were using. These were claims that I recall vaguely. You can probably expand on that, but it's come a long way from that day.
It definitely has, because with OJ, they were doing two different types of testing. RFLP, which is restriction fragment length polymorphism, that hasn't been done for many years. But with that type of testing, you needed to have a blood stain about the size of a quarter to get a DNA profile.
Wow.
Now you can have a blood stain that's, basically invisible. You don't need to even see it to be able to get results. So, and it couldn't be degraded DNA. DNA could not be broken down at all, or you wouldn't be able to get a result. So that's why when they introduced PCR, that's polymerase chain reaction, that's actually amplifying or copying the DNA. And now, again, that's why you can have just a few, really you want to have about 100 cells is a good amount, 150 cells is a good amount, but it's even less, really. You can get results from much less than that. So
that is something else. And now, and this goes into something now that you're doing again. You had left the lab. I think you had worked for a government agency for some time, for quite a few years, I think. And then you left the lab and you've done mostly consulting for a few years now, but you just recently opened your own serology lab. Is that correct?
I did, yes, very recently, just this month. Yeah, so I'm super excited to be back in the lab. So I'm not doing DNA testing, but doing serology. And again, that is body fluid identification. So currently at my lab, we're testing for blood, semen, saliva, and then 2 other tests that we're doing, which not many laboratories have, are for vaginal fluid, which again, many labs do not have. There's a couple around that I'm aware of. And then the other is menstrual fluid, which can be important if you have a blood stain and you need to find out, is this blood from an individual who is menstruating and that's why there's blood present, or is this, circulatory blood where they've been cut and are bleeding? So
and you can distinguish between the two.
Yes, yes. Well, it's considered a presumptive test, which so it's not specific just to menstrual blood, but it gives us a very good indication that it's more likely to be menstrual blood.
Interesting.
Yeah, and with the vaginal fluid, that test, again, it's presumptive as well, but I'm doing 2 different tests to help determine if vaginal fluid might be present. And the reason I can tell you that this is important, because I often get asked this on the defense side of things, where let's say it is a digital penetration case, and the laboratory, the state lab, is able to get a DNA profile from the suspect's hands, and they do have the victim's DNA on the suspect's hands. Well, then the question is, well, did the DNA get there because they, back to shaking hands? Is that how the DNA got there? Was it just casual contact? And if you're able to test for that and say, either yes, there are vaginal epithelial cells here, or no, I don't see any evidence of that, that can be really important information in those type of cases. So that's one of the reasons I really wanted to offer this test. Yeah,
interesting. I'd like to, we've got Mitch Jackson in the house, and we've also got Tamara Shelley. Tamara's an investigator, Mitch is an attorney. I don't know if either of them are available, but if you are and you'd like to jump in, Mitch or Tamara, please grab a seat. Here comes Mitch. Hi, Mitch.
Hi, you guys. How you doing? Great discussion. Great conversation.
Good.
Just fast.
Tamara, I don't know. Or, I'm sorry, Suzanna, I don't know if you, Mitch and I did an interview probably about a month ago. I don't know if you saw that or had a chance to view that. We did it through Blab, but he's an attorney down in Southern California, and he's got great stuff.
Okay. Hi, I'm here too. I'm in Carlsbad. Yes.
We used to race motorcycles down in Carlsbad back in the day. So, my thought on this is, there's Tamara. Hi, Tamara.
Hi,
Tamara.
Is, when I primarily try civil cases, and the DNA aspect of what you've been talking about doesn't come into play too often on some of my criminal cases, it has. But having said that, when we're picking a jury, a lot of jurors are predisposed to think about a topic based upon what they watch on TV. And on TV, what happens is somebody shows up on a crime scene, they find the DNA sitting right in front of them on the floor, whatever it is, and within 45 minutes, the crime is solved. In the real world, how long does it take to once you locate the DNA at a crime scene to when you're able to, come to some type of definitive finding, whatever it is you're looking for? Is it hours, days, weeks? I'm just curious.
Currently, to actually get the profile, it's about, you could get results within about
No,
a
you
day. You know, you if you worked kind of straight through, you know, you could get results within about 24 hours. Then the results have to be reviewed, et cetera, before it's released. That's not the norm, though. You know, the norm is you get a case into the lab, it sits there for 3, 4, 5, 6 months. When, you know, I mean, because you have all these other cases in front, right? So it takes some time. Now, in the future, and actually currently, there are some labs and some police stations that are using rapid DNA. So rapid DNA is basically a all-in-one instrument that it's mostly for reference samples. So it's going to be, I think it's in the next several years, you're going to see it at a lot of police booking stations. Just like getting your fingerprint, it's going to be, all right, buccal swab, and all the people have to do is put it in this instrument, and within 90 minutes, you can have a profile that's uploadable to CODIS.
Yeah, I thought you were going to tell me it would take weeks or months.
Yeah.
So that's fascinating.
It really brings up another question I had too for you, Suzanna, was I had been reading in the ABA Journal, amongst others, recently that in many states there's a backlog of rape cases or a backlog of DNA, and what you're saying now is that they're also the DNA analysis is also being applied to lesser crimes. So how do we how is that going to work out? Is that why they're coming up with faster testing situations?
Well, I mean, the faster testing is, like I said, it's going to be for reference samples, non-mixtures, things like that. You know, and so labs do have to wrestle with this, right? They want to do as much as they can, but sometimes you have to put a limit and say, we're not going to be able to take those burglary cases because we need to work on the current cases, or we can't work on cold cases because of anything else. But what sometimes happens to help alleviate that is government grants. So there are a number of federal grants that are specific for example, the rape kit backlog. There are grants that people can apply for. They can get funding for additional analysts or cold case grants. They can use that funding for investigators as well as laboratory analysts. So that can help alleviate some of the backlog. But you're right, there's so many items that you can get a DNA profile from that if people keep, you know, you want to maximize the potential of DNA testing, but you have to also make sure those really serious crimes, you know, don't get pushed too far in the backlog because you're doing the less serious crimes. I mean, anybody who's a victim of crime, whatever it is, that's serious to them. So that's kind of that's a hard thing to decide who gets the testing done first. You know? Very
interesting. It's a fascinating topic. And did you have something else, Mitch? I was also going to have Tamara maybe ask a question. You're welcome.
Oh, absolutely. But I would love for Tamara to jump in and share her thought or question.
I have all sorts of questions, but just a quick one before I ask the real one that I was wondering about. So are you just for hire for law enforcement investigations, or is your lab hireable for private investigations and private citizens?
You're kind of cutting out a little bit, but I think that you asked whether my lab was for hire for both privates or police investigations. Yeah, anything. It's anyone. Like I said, currently a lot of my work now is with defense attorneys doing consulting work. I'm still going to be doing consulting work. If they have particular cases that they need serology testing on, that's great. I'm happy to do that. But I also feel like with the lab aspect, there's also a need, you know, for, like you said, private investigators, police investigations as well. I mean, I'm not gonna discriminate. If you have a case, I'd be happy to work it.
And we've discussed it before that, you know, that's important to your credibility as an expert witness doing both sides. Yeah. Absolutely.
Yeah. Sure. And, you know, I mean, that's the thing. I come from a lab background where I've worked for the Florida Department of Law Enforcement and Charlotte-Mecklenburg Police Department, but I've also worked for a couple of different private labs. And so then, you know, private labs are going to accept, again, defense work or police case or prosecution work. And so it gives you I think it's great because it lets you see both sides of it. And I'm glad I kind of have that experience from both sides of it.
Can I ask one more follow-up before I have to bounce out, Nick?
Yeah,
Is that okay?
yeah, sure.
I'll be a second. Susanna, look, and I think Nick brought it up at the very beginning of the show, the O.J. Simpson case, okay? And I know technology has advanced substantially since that case was tried. But looking back on that case, looking at if that same case was going to happen today, God forbid, but something like that happens, what different approaches would DNA experts use today that weren't available back then? And the second part to the question is, in court, what is it? I always think lawyers do make a huge difference in the outcome of a case. In other words, the DNA could be great, but if the lawyer presenting it doesn't present it clearly, cross-examination, things don't go well. So maybe if you could talk a little bit about that with respect to DNA being used in civil and criminal courts, and then what weight in the outcome of the case do you believe good DNA has when you look at all the other factors that come into play? But my first most important question is, today's world, an O.J. Simpson type of case, what would be different? And I've got to bounce out, so I'll listen while I'm watching, okay?
Thanks, Mitch.
Okay. Thanks, you guys.
Sure. Okay. Thank you. Well, first I would say one of the, if you want to call it a good thing, I suppose, one of the things that came out of the O.J. Simpson trial was that it created greater standardization for laboratories and greater accountability. So after that trial, labs started realizing we need to be more, we need to have accreditation, we need to be following standard protocols, and so kind of helped with the labs, the DNA labs, become more standardized. And so that was one of the good things, at least in our field. Today, though, I mean, I guess the difference would be that you can get results from much smaller amounts of DNA. There could have been any number of DNA or evidence items that you could get DNA from that wouldn't even been looked at that time. And we see that a lot when I look at cold cases now. You know, with cold cases, I always say, listen, you might have already sent it to the lab, but there doesn't mean that that's it. You know, you might have sent it and you got no results or something like that. Now you can do additional testing. One of the things that I have in my serology lab is an MVAC. It's a wet vacuum system. It is great for collecting trace amounts of DNA. So this is something that's very new. You can get results even if something has already been sampled. Let's say you had a piece of clothing that someone had already swabbed or taken cuttings. But if you've already swabbed it, you can go back over with this MVAC and actually collect additional DNA. They've already shown that that works, that you can get results even if something's already been sampled. So I would say, and today, you know, if there was an O.J. Simpson-like case today, I think that the number of things that might potentially be evidence would be so much greater. And again, you wouldn't have to worry about RFLP. Mixtures also are a little bit, I was going to say a little bit easier to interpret, but that's debatable. It depends, because now we see actually more complex mixtures sometimes with the trace amounts of DNA. But on to his other point about DNA in the courtroom, yeah, I really think that a good defense attorney or a prosecuting attorney who can explain DNA, make sure the jury understands it. You know, you need to have the right person on the stand that can make something that is kind of complicated, make it understandable and explain, you know, like I said, many times when I testify now, my role is not to say, oh, the lab totally screwed up and they're terrible. I'm here to say, you know what, I agree with the results, but let's talk about what are some other possibilities for how this DNA got here? Is it only how the prosecution alleges? Not necessarily. That is one possibility, but here are some others. And so you have to have someone to explain that. And if the defense attorney doesn't know enough about DNA to know that there are other possibilities, then that, you know, that can make a difference in a case.
Tamara, did you have, you had another kind of main question. I'm not sure we got to it or not.
It's really case specific, but we
Don't
had
give
a
away
case,
any confidential information.
right? So we were investigating a case for this woman, and she was kind of wishy-washy on what she wanted. She wanted all this stuff, but she never wanted to put the money up and actually go through with it. I think she didn't really want to know, although she acted like she wanted to know. But she thought that her husband had fathered a child with another woman, and she wanted us to get a DNA sample and test it against his. And we were discussing this idea. She never did have us do it, but we were discussing the idea of how are we going to get a toddler's DNA from a kid that we don't have access to, and we don't have access to anything except maybe their garbage. And we were asking one of the local labs here, what kind of samples would you be able to get DNA from? Because we would have to determine that whatever we got actually was from the child. So we were thinking immediately a diaper or a sippy cup or something like that you knew that it was the child's and not the mother's or someone else's. And they were saying, the way I understood it is anything that is a bodily waste of any kind is not DNA. It won't reveal DNA. But if you're saying that skin cells have it, then that kind of makes me question what is a good sample of DNA, or what can it be drawn from?
Right. Okay. Well, yeah. The bodily waste, probably not the greatest ones. You can, there have been instances like burglaries where people go and like defecate in the corner. You can, if you swab it, it's the outer portion, you can get a profile. And urine, there is some DNA, but you would have to concentrate it. You couldn't just swab a little bit of the urine. There probably wouldn't be enough cells. So other than that, you would need, a lot of times with missing persons investigations, we say, well, a toothbrush, a hairbrush. You can get DNA from hair, but it has to have the root. So I guess you don't want to go up and pluck hairs out of a child's head. But if you had a hairbrush or something like that, anything that anyone has drank from. So, a cup, if they chewed gum, I don't think a toddler would be chewing gum, but, for other cases, chewing gum, anything they ate. Let's say the child was eating a piece of pizza and left part of it behind, and you were able to collect that from the garbage. You could probably swab the edge where the person had eaten from. That's been done in a number of cases. So anything that you can get saliva, that's the best, would be saliva, something that has saliva on it. Because
I was just going to say, you mentioned about 4 or 5 kind of main bodily fluid, maybe even 3 or 4 that are the best for DNA collection earlier. It sounded like saliva, some semen, and were there 1 or 2 others?
Blood. Yeah, typically it's blood, semen, saliva. That's what those are the, any kind of body fluid generally has a lot of DNA, so that's why they're good sources of DNA. When you get to things like touch DNA, then you don't know, you might get a mixture, you don't know for sure if it's from that person, so that's why that's a little bit not the greatest. If you're talking about an older individual, if you could get, an envelope or a stamp or anything like that someone had licked or even touched on the envelope, because it has that, if it's self-adhesive, let's say, and it has that sticky part, then skin cells would stick to that and maybe enough to get a DNA profile.
That is interesting. And Malia, who's over in the chat right now, had posed a question. She says, we've heard a bit about sometimes inaccurate DNA results. And could you elaborate on that? Now, I think that's maybe a little bit broad, but maybe you could tell us why in the news we'll hear about occasionally. What are the most common reasons we might get some inaccurate results?
I'm going to jump off, but I'll be in the room.
Thanks, Tamara.
Thanks.
Talk to you later.
Sure. Thank you. Inaccurate DNA results.
Are there some common reasons that they might be inaccurate? And I like to remind people that when this happens often with the law, and it probably happens also with forensic science, that when it's reported on, the they seem to glaze over certain things that somebody who's in your position would say, well, they're not telling us the whole story because they couldn't fit that in an article or something like that.
Right. I mean, it could be when you have inaccurate results, that basically to me indicates that something has been contaminated. So let's say, and this has happened in a couple of cases where an analyst is examining an item of evidence, they get a profile, they upload it, and it hits somebody in the staff database. Maybe they've contaminated it themselves. So that's one that'd be obviously inaccurate if it's their own DNA or someone else in the lab's DNA on the item of evidence. It could be a sample switch where, that's something that I always look for when I review cases. Unfortunately, you can't always tell just from looking at notes that something like that could occur without people noticing, without the proper documentation. So that's something that would be inaccurate. If you had two reference samples, and instances of this have happened. You have two male reference samples, and those get switched, and one of them matches the crime scene evidence, but you've switched the reference samples. So now you have the wrong individual that you're saying it matches. So, that can happen. But typically, so those are like some pretty gross negligence, or not negligence, but gross examples of, major problems. Inaccurate, there can be issues with mixture interpretation. So that would be something where maybe one individual at a lab says, yes, this person, I compared this suspect to the mixture and he is included. He's part of this mixture. And if you have someone else review it, they say, I don't know about that. I'm not sure if you can include him. So that's an interpretational issue. So that would, again, can impact.
And you kind of made mention earlier that the testing has become more standardized in the last, probably even in the last 10 years. Is there a federal accreditation or federal guidelines on DNA testing laboratories, or is it a state by state?
Yes. Any laboratory that is accredited. So, and you have to be accredited in order to upload profiles into the National DNA Database. So basically all the public laboratories and the vast majority of private laboratories are accredited. Although an important point is private laboratories cannot upload profiles into the database, even if they're accredited. So, but yes, if you are a public laboratory, the vast majority are, maybe all, are accredited. And so then that means that you have to follow the FBI's quality assurance standards for forensic DNA testing laboratories. So there are a set of standards that, go through everything, go through your facilities, your personnel, your procedures, and make sure. So not all the procedures are exactly the same. They just say you have to have procedures. They have to be validated. So there can still be variability. And again, with the mixture interpretation, the federal standards are not going to say you have to do it this way. They're going to say, here's some suggestions. These are our, but here's some things that you should probably try to do, but there's still going to be a lot of variability. It's not going to be one protocol that every lab follows. So each lab has their own protocols that have to be validated and followed by the personnel in their labs. And that's another thing I look for. Does the lab have protocols that follow the national guidelines, and are their analysts, are they following those guidelines? Because sometimes you can see, we call it protocol drift, where someone starts doing something a little bit different, and then the next time they continue on that difference until they're not following the protocols anymore, and nobody seems to catch it. So that can happen.
And that's okay. That's what happens with live streaming. That's
right.
There was another question from Tamara. She asked one in the side here. Let's pull that up. What might get a DNA result? What would cause a DNA result to become inadmissible? And there's probably several reasons, I'm assuming, on that.
Sure. I would say One reason might be chain of custody issues. If you don't have the proper chain of custody and you cannot show that you had control, or you knew who had control of that sample from the crime scene all the way through the analysis. So that's certainly one way that it's inadmissible. It could be that it's a DNA profile that is incomplete. So it's a partial profile, and perhaps the statistical conclusions are so weak that the judge says, you know what, this is going to be, it's no longer probative, it's prejudicial, where you've got, a 1 in 5 or something like that. But typically, I would say most of the time, even if it's very common statistics like 1 in 5 or 1 in 10, it still is allowed in. That's typically what I see. And then they say, well, the jury has to put weight on that evidence, or realize that there could be someone else that could also match.
Interesting. I'm going to invite, if you don't mind, one more person to come in and ask a question, if she's available. Malia Probst is loving your comments here, and she's really promoting your stuff, and she wants to... She wants to steal the chain of custody comment and use that for everything. So Malia, if you're out there and you'd like to come in, she's great. She's got a virtual reality blab once a week, and she's really, really into VR, and has now gotten me into virtual reality as a result of. Oh, she's heading off to the gym. She can't. Okay, Malia, no problem.
Oh, okay.
We understand. And I see Nancy Maryland's joined. Nancy, who is a legal marketer, works with attorneys a lot. Maybe Nancy wants to jump in. But I am going to ask Malia's last question here. So we did kind of establish, you did kind of explain that there were national protocols, but that there was some variables. And how enforceable are these protocols? And in my mind, I'm going to attach a little something here. I was thinking about these guys who are getting, expungements and so forth, who have been convicted, and then later on they find that some testing was done incorrectly. Is there civil liability for a laboratory in those cases?
Yeah.
Is it limited?
I can't. I don't specifically know. I know that the city or the county or the state can be sued. That has happened where it's in the exoneration. Is that what you're talking about? Like the Innocence Project and people are exonerated. Yeah. There can be definitely some civil liabilities with, typically, I haven't seen a lab that, that has, it's more the county or the state, or maybe the police agency
pockets,
or something.
They're going
like
to go
that.
for deeper pockets in the counties. And I can tell you, I can comment on that part, that they'll go for the, I don't want you to comment when you're uncomfortable. That's, and how enforceable are those national protocols? I would.
Well,
go ahead.
Yeah. You can't. Here's what would happen. Let's say you had a lab that was not following national guidelines. Then what happens is the laboratories are audited every year based upon those national guidelines. 1 year, it would be an internal audit. So your laboratory personnel yourself, you would go through and say, okay, are we following all these particular standards or not? And, are there anything where we need to update our protocols or whatever? So that's the internal audit. The following year, you would have an external audit where there'd be an agency that would come in. It could be another DNA lab. It could be a national organization that will come in and make sure you're following the guidelines. If they find problems that are not corrected and are very serious, then what can happen, and this rarely happens, but it has happened recently, is the lab loses their accreditation. If you lose your accreditation, you can no longer upload profiles to CODIS. You have to, so then you would have to make a lot of remediations to be able to start having access to the DNA database again. So, for example, the Washington, D.C., the laboratory in D.C. recently, and I don't want to say they lost it, but they closed the lab down. And it was, I think it was mostly having to do with mixture interpretation. So again, they had all the protocols, so they were following the national guidelines because they had procedures, but they didn't necessarily the procedures maybe weren't going far enough, or in explaining things enough, or they were having problems with their analyst following. I'm not precisely sure, but they did close the lab. So that would be ultimately what would happen, is they would either close the lab, or the lab loses accreditation, has to regain their accreditation before they can enter profile.
So, okay, and that makes sense. So we don't, if you guys aren't following this, we don't want you uploading your information to CODIS and
Right.
possibly tainting the bigger pool of data. It's what it sounds
Sure,
like.
sure.
And is there a regulating, Tamara had asked, who regulates the labs and accredits them? Is there a oversight sort of, or an association?
Yes, there's a couple. So what I've mostly been talking about is the FBI's guidelines. They have they have a quality assurance standards. However, there are other organizations. One is ASCLD Lab. That's American Society of Crime Laboratory Directors Laboratory Accreditation Board. So that's one that has been very prominent for many years, where they will go in and accredit the laboratory as it would be not just DNA, but let's say it's a crime lab that has firearms and they have chemistry or trace, whatever. They would accredit the lab all of those different sections of the laboratory. And that you have to be reaccredited every 5 years. And then there's another organization that also does that, that will perform the accreditation. So there's only two that I really know of that do the national accreditation. And again, that's every 5 years.
All right, great. And so we've got about, I had scheduled us from 2 to 3. So I usually kind of about this time, I like to ask, where do you see, kind of a two-part question, is what would somebody who's going through school to do what you do now, what kind of advice would you give them, sort of friendly advice, and where is, and to become an expert at your level, and where is forensic DNA analysis, forensic serology? What will we be looking for in the next couple of years? Advances and things like that. So what would you advise somebody in school with today?
Right. I would say, well, first of all, if you want to go into forensic DNA, then you need to have a natural science degree. So you need to have a bachelor's in either biology or chemistry or something like that. There are a lot of universities now that have a specific forensic science program. That wasn't really around when I went to school. I would say maybe take that as a minor and have the regular biology or chemistry degree, just because when you get out of school, if you have a specialization that's just forensic science, there aren't that many jobs. It's hard to get into this field. So if that's all that you are now qualified to do, that, you've kind of pigeonholed yourself. So why not get a biology degree or chemistry, and then you can do many, many other jobs while you're waiting for that opening at the lab. And also be willing to travel, be willing to move somewhere else. Because if you're like, I have a forensic science degree and I want to work in Pennsylvania or, whatever specific state, good luck. You're going to be waiting a long time.
It has to become more competitive because
so much more because of CSI, basically.
That's okay.
You can thank CSI for that. And, it's good. People were more aware. I got into the field before CSI, but you were talking about O.J. Simpson. That was one of the reasons I became interested, because I was taking a genetics class in college at the time. And I was like, oh my gosh, there's something that I'm doing in school is actually in the real world. So, I feel like you learn so much stuff that you're never going to use, that it was fascinating to me. So that's kind of what pushed me in the direction. But so with CSI, when I went to school, I did go to grad school. So I have a master's degree in forensic science. But when I went, there were like 10 schools in the country that had master's degrees. There were no undergraduate programs. But now there are so many, and there are so many people that are getting into this field that maybe don't really know what it's all about. They see CSI or NCIS or whatever and think it's a little bit more glamorous than what it is. And I guess that's my main advice, is I would just get a regular biology or chemistry, natural science degree, and so you have a lot more opportunities.
And
what was the second
I was just going to actually add to that. In your practice is been much more clinical than it has been what you see on TV, right? Much more laboratory driven than, yeah.
Yes. Yeah. I mean, I think maybe people are more aware of this now, where they know that if you're a CSI, that doesn't mean that you're going to the crime scene and you're carrying a gun and you're arresting people and then you're going back to the lab and getting the results. No, pick one. You're not going to do it. Those are like 5 different jobs in one. So it just doesn't happen. And frankly, I think that if you have that science background, I mean, I don't know. You're going to be so busy in the lab. Like, for example, when I worked for the state of Florida, I really wanted to cross-train on crime scene investigation. I really wanted to go to crime scenes. I wanted to be trained on it. They said, sorry, we need you in the lab. We've got too many cases, too much of a backlog. We can't have you going to do that. We need you here in the lab. So that's kind of one of the downfalls of watching CSI. I mean, you can't believe
everything you see on TV. And kind of the second part of that question is what is the future of this forensic DNA? And I from what I see from our members on Experts.com, including yourself, is that the science appears to be getting better, and even some sciences that used to be considered strong are now being eliminated because of the advancements. They're seeing, look, that we can't rely on that any longer. What does the future, the next 5 to 10 years hold for forensic DNA analysis and forensic serology in your best estimation?
Well, I would say we're going to see probably, hopefully, I'm hoping, more body fluid identification. That is, maybe not what I'm currently doing. So I'm doing the kind of the more conventional, traditional forensic serology. I see it going more to mini or micro RNAs. So instead of looking at DNA, looking at RNA and how the proteins, how things are transcribed and how that relates to what body fluid might be present. So right now they're not commercially available, but many people are doing research to say, if you test something, you can tell whether it is sweat or urine or vaginal fluid or, just from a very small amount of material. So that's something I see in the future. And then the other thing that is probably going to take longer is there's a completely different way of analyzing DNA called next generation sequencing, or NGS. And it's going to take a while to get into the labs because it's a different platform, completely different, from what they're doing right now. But the benefits are that you can take a very small sample of DNA and you can get tons of information. You can get your regular results that you're used to seeing now that are uploadable to CODIS, but you can also get many, many more loci or areas of the DNA, not just the 16 that we look at now. You can also get Y STRs. Y STR is found on the male chromosome only. So we can do that now, but then that's 2 different tests. You can get, your regular profile and then Y STRs. It does it all at the same time, and forensic phenotyping. So phenotyping is using DNA to find out information about how the person looks. So right now we can tell if you're a male or a female. That's it. All right, so now it's going to be, and there are already labs that do this particular part where you can tell the ancestry of the person, so their genetic heritage, their Caucasian or Hispanic, things like that, eye coloring, hair color, whether they have freckles or not, and so that's going to, I think, increase, and you're going to get more and more of that. So that next generation sequencing I was talking about, that also incorporates that. Pretty cool. So, and there are labs, there's Illumina is one here in Carlsbad, that they have a kit that can do all of that. They have the instrumentation that can do all of that. It's just waiting for the standards to be, waiting for the labs to validate it, the standards to be written, and making sure that it's acceptable into CODIS. Wow. And then
that's fascinating.
It's going to take a while, but it's coming.
That's fascinating. Yeah. When you think about the ability to respond quickly, or if there's ongoing investigation. You think you have somebody who may strike again or may do something again, being able to identify hair color and male versus female and those things. I don't know how rapidly that could happen, but again, this was what we might see in the future. I mean, it seems like that would be incredibly powerful information to have.
Right. And again, some of that testing is currently available. So let's say you have a profile, you have a male profile from a crime scene. You have put it into CODIS. It doesn't match anyone. You've compared all the suspects you have. It doesn't match anyone. You can send that to, there are, again, I think there's 1 or 2 private labs. One that I'm thinking of is called Parabon, and they can use that sample and do their testing and tell you a lot of information like hair, eye color, genetic background, things like that. So ancestry.
That is fascinating. That is great.
Yeah.
Well, we are about 5 minutes out, but I got to tell you, I'm going to say it while we're still on the record before I pause the recording. We have had a far superior response than we've had up to 50 people in the room at a time watching. And, so I want to thank you for that. Thank you for taking the time. Sure. I think I wouldn't be surprised. Maybe we can get from I think we wouldn't be surprised if people would like to hear more about you in the future. Can we get ones if you would like me to have Suzanna on again in the future and maybe more of a specific topic? Yep. And Tamara's paying attention. Jackie.
I'd be happy to come back. Everybody loves DNA.
It is. I mean, it really is a fascinating topic. I was why I was really enthused for this, and you are just you're great to work with because you were like, let's do it, and so I like that. Sure. Is there anything? So let's tell people before we wrap it up, before I stop the recording, where they can find information about you. I posted a couple links earlier, but why don't you say it?
The best way to get the most information would be my website, which is www.ryanforensicdna.com. And check back because I'm getting a brand new website. It's probably going to be ready tonight or tomorrow, so it's going to be totally different. You can look at it today and then make the comparison. So it'll have more information about the serology testing that I'm talking about on that website. And then, I have my, obviously my email address. So if people have specific questions, I don't mind taking a few minutes here and there to answer some questions too, if you want to email. But the best way to get information is my website. You can sign up for my newsletter on my website, which initially I tried to send out every 2 months, but that wasn't happening. I'm pretty busy, but when there's something really interesting that intrigues me, I like to do a little more research and then write an article about it to try to help explain it. So there's a few back issues.
Excellent. Well, thank you very much. And she can also be found, Suzanna can also be found on Experts.com. So I've posted her website in the chat bar, and this is a link to her profile on Experts.com as well. And any last minute thoughts you have? What did you think about your experience on Blab for the first time? Do you like how this works? And it was really, I thought everything, was really smooth and just, I liked it. It's interesting. I mean, one of the things I really like is talking to people about DNA. So, I wish that I had a chance to answer more questions. So if we do this again, I definitely, because, I know what I know about DNA, but I don't know if I'm explaining it properly or if there's particular nuances or questions that people are like, well, wait a minute, so that helps me really, I think, to know what I need to do to explain things better. And I think, maybe in the new year, once I get a new calendar for scheduling on these, I think I'd like to have you back because I know that there were questions that we were unable to get to in our timeline, and I'd like to bring some more people in with some of those lay questions. I think that would be helpful. So if you're up for it, I think it would definitely be informative. So. Again, I'm Nick Rishwain with Experts.com, and we were joined, my guest today was Suzanna Ryan, and she can be found at ryanforensicdna.com. Thank you for being on, Suzanna.
Sure. Yes, thank you for having me. This is a great program that you guys set up. I'm very